This is a brief description of our novel tracing method for image stacks with globally varying contrast. Neuron tracing is the technique for extracting the centerline and underlying structure of neurons. It provides the model attribute of the neuronal morphology. Tracing method can be applied to any curvilinear structure such as networks of roads and retinal blood vessels. Our focus is on neuron image stacks with insufficient contrast and spatially varying signal-to-noise ratio. We propose two methods for neuron tracing in a single image stack based on open snake population. Our main contribution is the cooperation among snakes to combine global shape with local features. The main difference between these methods is the problem formulation. The first method formulates the problem as the joint probability optimization problem over a set of snakes S and mask volume Y given the input data X. The image stack is split in two box subvolumes to handle the varying contrast. The bottom right shows the tracing process of our first method in both 2D and 3D. Different colors represent different snake. These snakes evolve simultaneously based on local image statistics, local shape smoothness, and pairwise attraction among snakes. The most important factor is the snake population prior PS. It composes of local smoothness and pairwise attraction models. The smoothness energy is the internal energy of active contour model, where the elasticity term prevents snakes from stretching and the rigidity term restricts snakes from bending. The attraction model is the sum of the attraction map produced by a pair of snakes. The attraction energy is influenced by three factors. The first factor is the distance from the snake. The further from the snake, the lower the degree of influence. Second, only snake endpoints create attraction. Last, the snake attracts only along the snake's orientation. In the second method, we model the snake population using the conditional random field framework. Our objective function is formulated as the conditional distribution over the set of snakes S given the data volume 10. Bottom right shows the tracing process of our second method in both 2D and 3D. Different colors represent different snake. The association potential A is the energy function of active contour model measuring the likelihood of a snake being neurite. Interaction potential I merges neighboring snakes based on four criteria. First, snakes must align towards each other. Second, they must be close to each other. Third, they merge only at endpoints, and last, they must have sufficient length. We pose the problem as a multi-class CRF model, where snakes are the sites. With the graphical model formulation, we can apply the efficient inference like the highest confidence first method, which provides the order in which sites are optimized. HCF chooses to optimize the site with minimum stability, which is influenced by snake's length, number of neighboring snakes, and snake's energy. Then, the configuration of the snake is updated by the greedy algorithm for efficiency. Here we show the samples of our tracing results in red color on the sensory neurons of larval Drosophila dataset, whose volumes contain spatially varying signal-to-noise ratio. Columns correspond to different image stacks. We compare our results against two algorithms from Diadem Challenge in two sample volumes. Top rows show the collapsed image stack, and the magnified region with the ground truths in red color. Bottom rows display the collapsed 3D traces superimposed on the collapsed stacks of two Diadem Challenge algorithms and our methods. Different colors correspond to different neurons. We can see the previous methods either fail to detect neurons or exclude the background, while our second method is able to separate the neuron from the background and recover the correct neuronal topology.